Now, just to let you know what's happening behind the scenes here. So I talked with Professor Ceballos yesterday, and so because this class goes zippity fast, as all of you are already experiencing. The material that you're going to be tested on is going to be, so basically I wrote the questions for my material so that what I talk about in class is going to be hopefully aligned with what kinds of questions you'll see on the exam, right? So, as you know, from your experience with classes here, each professor has their own way of doing things. And so just to make sure that, that my style is what you'll see on the exam, I actually wrote all the questions for my part of the exam, which like you have like in no time at all, right? Thursday, like that next week. Yes, I know. Wow. All right, so just let's let you know so you keep everything in order. Okay, so because my style might be a little bit, as you'll see, my things I'd emphasize might be a little bit different than what you get in other classes. Okay, so but that will be reflected on the exam, and I'll try to prep you for what you might expect. Okay. So there's a copy of the lecture notes, and I think Professor Ceballos posted on B2L for you. Okay, and so I'm going to get started. What we're going to talk about today, we're going to talk about metabolism, and this is important because we're going to be addressing this in the lab, some of the lab experiments that you're going to that are upcoming next week. And so this will tie into some of the metabolic processes that you're going to learn about. You'll actually get a chance to see for yourself in lab. We're going to talk about, some of you know about this, the central pathway of metabolism, so glycolysis, Krebs cycle, electron transport chain, all those things that you just love. Right? We'll address that between now and Monday. And something that I do with my students, and because I'm the teacher, I get to do this with you. I like to have problem-solving exercises during class, so I actually have one for you today. And I'll explain more about this. And Professor Ceballos gave me permission to actually grade them and give her the scores, so I'll be doing that and returning those to you on Monday. But we're going to do that. We do that with my students. We do that each day, and so I'll say a little more about this. I don't know if she has done this with you already, something like this. Okay, so.
Yes,
all right. It just gives us a chance to see how you're doing with some of the material, especially now that we're on this accelerated schedule. Okay. Yes. All right. So you want me to stay in one place, or?
You're fine.
Okay. All right. So I like to start out with the basics here, and now some of this is going to remind you of chemistry, and hopefully all of you taking something like 305. How about 306? Anyone taking 306? You? Okay. All right. So you'll see a lot of this reflected in this. See a lot of that reflected in the next few sessions here. But I'm going to start with some basics here. So we're going to talk about metabolism, and these are chemical processes that are carried out by living organisms. Now, in a class like this, we're going to emphasize microbial metabolism. Okay? And that's actually since you haven't seen me before, that's actually my specialty. I do research with microbes, especially E. coli. And so we spend a lot of time looking at the metabolism of E. coli. But these are just chemical processes. And what you'll find is a lot of the processes that occur in E. coli also occur in your cells. Okay, so you can see a lot of parallels. And in fact, in our field of study, we actually use E. coli as a way to understand more complicated processes in humans and other organisms. Now, there's two basic parts to metabolism. One is called catabolism, the other is called anabolism, or biosynthesis. The way to look at this is that in catabolism, we're just looking at complex molecules. You're taking big molecules, and you're breaking them down into small molecules. And when you do that, you get energy. Okay, that actually releases energy. And we're going to see how cells can take advantage of this chemical process in order to get the energy that it needs to survive. Okay, and I'll go into that a little bit more. Now, the opposite of this is anabolism. So that's the way to look at the relationship between these two. Okay? Metabolism, you take big things and break them down into small things. In anabolism, you take small things, small molecules, and you build them up into more complex molecules, right? Now, in order for this process to happen, you actually need to add energy. It requires energy. Just like if you were to build a house, or build a computer, build something, right? It actually takes energy to do that. Right? And so the same thing occurs in cells. Right? You actually need energy to be able to do this. Now, a lot of you know this already, so when we talk about cells, what's the energy that we're actually using? ATP, right? So you're going to see that pop up throughout this chapter. Okay. Any questions about this? All right. So again, feel free to ask me questions anytime. Okay. Make sure you learn this. All right. So let's go into ATP. Right. So ATP. This is the major energy carrier of the cell. We like to look at it in terms of energy currency. Right? This is the main currency that cells use for energy. And the way it's stored are in phosphate bonds. And so I have a picture here of ATP. Okay, so here are the phosphate bonds, the phosphate groups, and here's the phosphate bonds. These are special bonds in that they store lots of energy. And so by breaking off one of these bonds, that allows the cell to be able to take that energy and use it for whatever processes it needs to do. All right, so ATP, this is what's used for the metabolic processes that require energy. So we looked at anabolism, that's where the ATP comes into play. And this is actually very important that cells have ATP. Now to give you a way of picturing this, I'm going to go back to E. coli. So if you look at one E. coli cell, now I think that by this point you actually saw an E. coli cell, or maybe many E. coli cells in one of your stains. Okay. So just take one of those cells, one of those itty bitty cells. If each cell contains 5 million ATP molecules, 5 million. Right? But in order for E. coli to live, in order for it to survive, it needs to use 2.5 million of those ATP molecules per second. So based on its store of ATP, how long will it last?
2 seconds.
Yeah, 2 seconds, right? Okay, what that tells us is that E. coli, in order to survive, it actually has to always make ATP. Right? So this process of making ATP, making energy, is a constantly ongoing process for cells. And E. coli is a good example of this. Right? So if it doesn't make energy, it's not going to last for very long. Right? All right, so we're going to be spending a lot of time in this chapter looking at how cells make ATP. And the way they do this, there's 3 methods. And so I'm going to try to highlight these methods as we come to them. One is called substrate level phosphorylation. Okay, so this is where we take phosphate from one molecule, from one organic molecule, and just simply transfer it to another. Right? So you'll see a few examples of that. Okay? So that's one way that you can make ATP, just take phosphate from some other organic molecule. There is another process, and we're going to spend a bit of time walking through this. It's called oxidative phosphorylation. So this is where we use energy, get energy that's released by transferring electrons. Transfer electrons from one molecule to another, and that allows you to make ATP. Now some of you may remember this, right? There's one process that does this, which is what? In the central pathway of metabolism. Electron transport chain. Okay, so that's where we're going to come across this, and we're going to take that apart a little bit to be able to show you how that works. Okay, that's where that's going to come up. And then the final one is photophosphorylation. So this is where we use light energy to make ATP. And so again, some of you may know where this occurs. Where? Yeah, photosynthesis. Okay. All right. So this is a way that we relate microbiology to the rest of biology. And we tie it into some of the processes that you may have learned in other biology classes. Okay. All right? So again, what we're looking at is how we make ATP. We're basically going through this process where usually we start with ADP. It has 2 phosphates. We add a phosphate, and then we have to have some form of energy to be able to make ATP. Okay? Yes?
Something I've never been exactly clear on, and since it's your area of expertise, maybe you can help me out. To make ATP requires ADP,
Yes.
another phosphate, and some energy.
Right.
And the purpose of ATP is to store energy. What is? Oh, wait, so she's I can't think. Okay. What is the ratio of the energy input compared to what you actually get? If it's close, then I don't see the actual point.
Yeah,
You understand what I'm saying?
yeah, so remember that often cells take in, so mostly a lot of the energy that a cell gets.
Okay, I'm sorry, if we could just call it units.
Okay.
How many units of energy does it take to make ATP, and then how many units of energy do you get out once that bond is split and the ATP molecule is utilized? In other words, is it 1 to 10, 1 to 2?
Yeah, so if you look at a sugar molecule, right, look at a sugar molecule and you see there's actually energy in that sugar molecule because of the bonds. Okay? And so after we go through all our processes that we're going to talk about, which is the central pathway, it recovers maybe about 20 to 30% of the energy. Okay, but remember that's free stuff, right? Because it just takes in that sugar. That's free. That's like free money, free currency to the cell. Okay, so that's just like getting a free tank of gas for your car, right? You don't have to buy anything to get that. It just comes to you. All right. So whatever you get out of this, whatever you get out of it is bonus to you. Right? Now, a lot of us think that 20 to 30% is not that, it's like a lot of waste, and it is. However, it's actually much more efficient than anything that we've been able to devise mechanically. So your car is actually a lot less efficient.
You understand what, I'm sorry, but
Yeah.
did you get what I was asking? Because that's not what I was asking.
Okay,
I'm asking, if you look at the amount of energy that is input to make an ATP molecule, how many more times energy, how many more times in terms of energy units do you get once it's split? Because if it's fairly even, then you're not getting anything. You understand what I'm saying?
Yeah.
So how many, just let's call it units, how many units of energy are required to make an ATP molecule, and once that bond is split, how many units do you get back?
Oh, that's actually very efficient. So that's because you're working at a molecular level, so that's going to be very, very efficient. So whatever energy gets put in, it all gets extracted out.
Okay, but then you're not getting, you don't understand what I'm saying? You're not getting anything out of it. In other words, it's requiring energy to make the ATP molecule. And the purpose of ATP is to store that energy and use it. But if you're not getting any more than you're putting into it, then I don't see the point.
So why don't you hold on to it? Because I think you have to put the question in the right frame. So let's take a look at that. Okay. All right. So again, what I'm going to do is go through some basics of some definitions with you to make sure we understand this. Okay. So when we oxidize, so we're going to talk about oxidation and reduction. So when you go through oxidation, what you're doing is you're removing an electron from a molecule, and that process of removing an electron releases energy. Okay, releases energy. When you reduce a molecule, you're adding electrons to a molecule, and in a cell, what that does allows you to store energy. So we're going to actually see this process occur where you're storing energy, okay, by actually adding electrons. And so what happens is that in biological processes, we find that these two reactions are coupled. Okay, they're coupled. So that is, if you oxidize one molecule, you're usually reducing another. So when you look at this example, so basically I represent the molecules of these blobs, an orange blob and a blue blob. If you take this orange molecule, you start here and you go here, which process are you going through, oxidation or reduction? Oxidation, right? You're actually losing an electron, okay? And the blue molecule is undergoing what?
Reduction.
Reduction, right? So that's what we mean by they're coupled, okay? All right, so we're going to see examples at a rudimentary level, at a basic level, about where we see this type of coupling occurring between oxidation and reduction. All right, so again, I'm going through some basic terminology with you before we head into looking at metabolism in more detail. So another molecule we're going to talk about are enzymes. Right? So an enzyme is a catalyst, and in this case it's a protein catalyst. All right, so these are substances that remain unchanged while speeding up a reaction. Okay, so they remain unchanged while speeding up the reaction. That is, you can reuse them over and over again. And they actually speed up that reaction. So when we look at this, first of all, enzymes, they require specific substrates. Now, a substrate is the molecule that an enzyme reacts on. Right? So we'll look at examples where it breaks down a molecule. Right? The molecule that breaks down is called a substrate. There are other examples where it takes two molecules and combines them together. Those two molecules, again, are substrates. Right? So it's just the molecule or molecules that an enzyme acts upon. Now, where the substrates interact with is called, with an enzyme, it's called an active site. So in an enzyme, there is an active site, which is where the molecule, or more than one molecule, will bind. Okay? So here's an example of an enzyme. So this is more of a cartoon diagram of an enzyme. So here's the enzyme, here's the substrate. That substrate will interact with the enzyme in these positions here, and that's called the active site. Okay, so this is where the substrate binds to the enzyme. Now, and this is a little more realistic view of an enzyme. So again, here is the active site. Here's the substrate. The substrate fits within that active site. And what's special about this active site is that it's very specific for the substrate. Okay? So this is where you get the term specific, why you'll see specific pop up. Enzymes react with only specific molecules. Okay? And that's what makes enzymes so powerful for a cell. It allows you to do metabolism with only the molecules that you want to be reacted upon. Right? Another property about enzymes is that they can lower the activation energy. So the activation energy refers to the amount of energy required for a reaction. So to give you a picture of this, I think I have one right here. Okay. So this is, let me just check to make sure it is sent here somewhere. It's on page 3. Okay. Here is a picture of a graph of what happens when an enzyme reacts with molecules to make certain products. And so we have it, the way that we have it graphed is this is the amount of energy that's needed, and then the amount of energy that's used, and then this is the progress of the reaction. Okay, so in a typical reaction without any help, this is the energy curve that you have to go over, the energy barrier that you have to go over. So you have to supply this amount of energy to be able to make this reaction go. Okay. What an enzyme will do is that it lowers this amount of energy that's initially required, the activation energy. Okay, so now instead of having to go across this hurdle, you just have to go over this hurdle. Okay, so it lowers the activation energy. Now, you may have done this in chemistry. There is a way of getting this reaction to go without using an enzyme. What's typically what do you do to a reaction to make it go?
Heat it up.
Yeah, you heat it up, right? Okay. Now, you can also do that with our molecules, but what's a problem with that?
It'll kill the cells a lot of times.
Due to degradation.
Yeah, right. So you can destroy a lot of things in that process. Okay, so enzymes are a way of making these metabolic processes more biologically friendly. Okay? And that's what's essential for cell metabolism, right? Okay. So here's an example. Here's another way of looking at an enzyme substrate reaction. So here is our enzyme. It turns out to be an aldolase. Here is the substrate. And then what happens is that the substrate interacts with the enzyme. Here's the active site. And then as a result of this interaction, you get these two molecules, okay, as your product, right? And as a catalyst, the enzyme is can be used again, can be recycled, and so now it can react with another substrate. Okay? Now, when you look at the products, actually just going back to where we started, when you look at the products and compare it to the substrate, is this a anabolic or a catabolic reaction? Catabolic, right? Because you're starting with a large molecule and you're making it into smaller molecules. So it's actually catabolic. Okay? All right? Any questions about this? Okay, so those are the different properties of enzymes and some of the different terms that we're going to use for enzymes. Now, something else about enzymes is that they often require help in order to work. Okay, so another term are so another set of terms. One is called a cofactor. Okay. A cofactor is can either be an inorganic ion or a coenzyme that associates with an enzyme to increases activity. So one way to illustrate this is here we have an enzyme, and in this enzyme, in order for this enzyme to react with the substrate, you need to add a coenzyme, okay, or let's say a cofactor, in order to make this enzyme more reactive with the substrate. Okay? Common example of a cofactor, one that's inorganic, is magnesium. That's used in a lot of different types of enzyme reactions. Right? And then a coenzyme, often they're vitamin-derived molecules, and we'll come across one when we look at metabolism called NAD+, so nicotinamide adenine dinucleotide. So that's an example of a coenzyme. And again, it's to increase the efficiency of an enzyme reaction. Okay. Besides microbiology, I also teach biotechnology, and if you don't know what that is, you actually cover a little bit of that in this class later on during the course. And we often work with enzymes that react with DNA. You'll also learn about DNA in this unit later on. And those enzymes require magnesium as a cofactor. If you were to actually leave out the magnesium, the enzyme would just be completely dead. It wouldn't work at all. So you really need those cofactors in order to make these enzymes work. All right, let me also tell you about some other things that can occur with enzymes. You'll be looking at some of these properties later on during lab, during one of the labs. So enzymes are also sensitive to the type of environment that they're in. So, for example, you could have temperature effects on enzymes. Right? So here we have a graph which shows what we're looking at is we're measuring the enzyme activity at different temperatures. Okay? And so now if you were to look at this curve, the one on the left, right, what temperature is it most active at?
38.
Yeah, 38, 37, right? Actually, does anyone know where you would find this enzyme? Where would be a good place to find this enzyme if it's most active around that temperature?
On your skin, on your body?
Huh? Yeah, human body, right? So you find this in human cells. Also, that microbes that live inside your body, they often have the same kind of temperature profile, right? So so what you find, this is very common with enzymes, is that as you increase the temperature, the enzyme activity goes up until it reaches a peak, but then after that, it drops off. Okay? Does anyone know why does it drop off? What's happening to the enzyme? Yeah, denaturing, okay? So this shows you an example of what's going on here, right? So an enzyme, just like other proteins, they normally have a three-dimensional structure. Right? So they actually have a defined structure. And here's an example. Okay? When you disrupt that structure, temperature is a good way to do this. You denature it. So what happens is, what that means is that you actually lose that structure in the protein. a good way to see this is if you take like eggs. So, you know, if you're taking an egg right now at this temperature, what would it look like? What would the egg white look like?
Clear.
Clear, right? Clear, runny, right? However, if you heat it up, right, it becomes that nice solid white, okay? That actually occurs because you denature that protein, right? And there's actually enzymes in that egg white besides what makes the egg white. There's other enzymes, and you'll find that those also get denatured along with the egg white protein. Now, you can also look at pH. So, again, we have a curve, we have a graph where we show the enzyme activity at various pH measurements. Okay. So in this case, where do you find most activity?
Neutral.
About neutral. Okay. Now, just make sure we have the pH straight here, because you actually have to do some experiments with pH in lab. Right. If you go down in pH, are you acidic or basic?
Acidic.
Acidic, right? So go up, you're basic. Okay, so you're going to see how metabolism can alter the pH of its environment in some of the labs. Okay, so again, enzyme activity is sensitive to pH and sensitive to temperature. Any questions about this? All right, let me give you another property of enzymes, and this actually gets into some of the applications for enzymes. There's a lot of clinical applications that you can use with enzymes, understanding enzymes. And one thing that you find in different types of, especially in pharmaceutical therapies, is this idea of competition, of competitive inhibition or non-competitive inhibition. Okay? So it's possible to inhibit enzymes through these processes. Now, what I'm going to do is at least describe it to you right now, and then we'll go into this a little bit further. So, in competitive inhibition, what you do is what happens is that you have an inhibitor, and the inhibitor fills the active site of an enzyme. Okay, and because it fills that active site, which is normally where the substrate binds, right, it actually competes with the normal substrate for that active site. Right, so you can draw it like this, where let's say you have, here's your enzyme, here is your substrate, which only binds there. You also have an inhibitor, okay, which will also compete with that same site, right? So this is an example of competitive inhibition, right? The other type of inhibition you can have is non-competitive. Again, you have an inhibitor, but in this case, it binds to a different part of the enzyme, which is called the allosteric site. Okay? What that does is that it causes the active site to change shapes and no longer binds to substrate. All right, so what happens in this case is that you have, instead of binding here, it binds to here, okay? And by doing that, that changes the active site so that it no longer can bind the substrate, okay? So that's how a non-competitive inhibitor works. And where we see this happen, at least metabolically, inside cells, is using a process called feedback inhibition. What happens is that when you look at a metabolic pathway, the last product often acts as an inhibitor in the earlier part of the pathway in order to act as an off switch, an on-off switch for that pathway. And I'll show you an example of that a little bit later on. Okay. All right, so just go over this again. So this is competitive inhibition, where we have a substrate, an inhibitor. They both compete for the same active sites. Okay. These are diagrams from figures from your book, from chapter 5. Here is non-competitive inhibition. So again, we have an active site, here, the inhibitor binds to a different site on the enzyme called an allosteric site. And when it does that, it changes the active site so that it no longer can bind the substrate. Okay, so that's what we mean by non-competitive inhibition. Right? And biologically, where you find these types of reactions is in feedback inhibition. So here's the metabolic pathway, and then what happens is, in order to turn this off, what some cells will do is they'll take that last product, let's say it's, and they use that as an off switch, which will negatively, or which will inhibit this first step. Okay, so here it competes. Here it inhibits this first step so that you stop this entire process. Okay, which actually makes sense for the cell, because if you have a lot of your product and you don't want this pathway to run anymore, you can simply use it as an off switch and turn it off. Okay, so that's what that's one use for for non-competitive inhibition or feedback inhibition. Any questions about this? All right, now we won't go too far in this class to go through examples of this, but one thing that you might find in other biology classes that you'll actually see some specific examples of where this process gets used. So let me give you a medical example just to try to nail this down. So here's a chemical that you may have heard about. It's called sulfanilamide. Right? Does anyone know what sulfanilamide is? Or what does it use to do? What do you do with it? What's a common name for it? It's medical. Sulfanilamide sounds like sulfa. Right? Sulfa drugs. What are sulfa drugs? Let me ask you this: what are sulfa drugs used to do?
Things like tetracycline used to kill bacteria.
Yeah. So you use it to kill bacteria. Right? It's actually an antibacterial medicine. Later on in the course, you'll learn about antibiotics. Okay, which is related to this. Right? Sulfanilamide is also another type of antimicrobial compound. It's used in medicine, so and we often hear about this as sulfa drugs. Okay. Now, this one particular sulfa drug, sulfanilamide, is similar in chemical structure to a biological molecule called PABA. PABA is converted to folic acid by an enzyme. So bacteria have this enzyme that converts PABA to folic acid. Right? Now, some of you know this just from your own cells, from your own biology. Is folic acid an important molecule for your cells? I hope so. Okay, it is. Okay, right? So, in fact, it can lead to, like, if you're pregnant, it can lead to birth defects if you don't have enough folic acid. Okay? And just like that, for bacterial cells, it's also an essential molecule. And so, sulfanilamide actually inhibits the enzyme that makes PABA into folic acid. So, if you use sulfanilamide to inhibit the enzyme, what's going to happen to a bacterial cell?
You're not going to make that
Will
die.
it live or will it die?
Die.
It'll die, all right? So you actually kill it. Now, getting back to this. So here's paraminobenzoic acid. This is PABA. And this is sulfanilamide, okay? The question is this: since we know that paraminobenzoic acid since we know this is a biological molecule, this is the inhibitor, and we know that this inhibits this enzyme, would this be competitive or non-competitive inhibition?
It's competitive.
Okay, competitive, and why do you say that?
Because the structures are similar.
Yeah, if you look at the chemical structures, they look similar. Okay, so in fact, it is competitive inhibition. Okay, all right, and so. One reason I bring this up is that a lot of the drugs, many drugs that we use, act by this process. Like they're similar to what's normally found biologically. And so if it's something that a bacterial cell needs or that a virus needs, right, if you can make that into an inhibitor, right, you have a way of targeting that cell for destruction, okay, which is good if you're trying to treat disease. All right?